So this is a catch-up and question and answer and review section. I don't have a PowerPoint prepared. I guess I was planning to be guided by your questions. As far as the problem set is concerned, I think you guys have me figured out pretty well. You all did very well. I took off points here and there for not answering exactly the question or including too many or too few significant digits. Actually, not too few because I made a mistake in the question, but too many I did take a point off for. Just because. But if you have questions about the problem set or anything, now is a good time to ask it. I did give full credit for the diuretics and for the acetazolamide, the Diamox question, even if some of the details that I was looking for, that I was expecting that you might see in my answers, which are on Moodle, I graded that generously. That is to say, I didn't look for exactly what I gave in my answer, as long as you had information that was correct. Would it be helpful if I simply left the room and let you sort of reach consensus about some of the things that you would like me to go over, and then some representative from the group says, Bob, we'd like you to talk about whatever? Would that be a helpful way to approach this question and answer section? I see one hand up, so someone's willing to ask a question, but I'm interested in knowing if there's a way to Gerald, if there's a way to activate the conversation. Let's just start with Claude's question first, and we can decide whether I should leave the room and confer.
I feel struggle with differentiation between the exchanger and transporter.
Okay. Struggling with the difference between an exchanger and a co-transporter. It's pretty simple. Here is a membrane. Here is a membrane protein. A co-transporter, like the sodium glucose co-transporter. So let's just be clear here. What cell am I talking about? Well, this is a membrane of a cell. Let's get oriented. Here's another cell with its membrane. So I'm drawing two lines for a membrane, whereas in the past I often drew only one. Hope that doesn't throw people for. Maybe I'll just. No, this is okay. So I'm looking at the sodium glucose co-transporter. It's in my mind. What segment of the nephron am I in? The proximal tube. By definition, I'm in the proximal tube. The sodium glucose co-transporter is called the sodium glucose co-transporter because it moves sodium and glucose together. Co means together. There's another transporter in the basolateral membrane of this and I think most cells in the renal tubule in the nephron that moves bicarbonate in exchange for chloride. I'm gonna make sure I'm doing this right, because I don't want to give you bad information. So I'm gonna go to a slide that I have in my mind, but I'm okay. So I guess I'm thinking over here. A chloride bicarbonate exchanger. So it's not in the at least in this figure, it's not listed in the proximal tubule, it's listed in the ascending limb of the loop of Henle. Okay, so let me clean this up. Now, talking about a different cotransporter. I'm going to use 3 arrows. What am I thinking of? Sodium, potassium 2 chloride co-transporter on the apical membrane, and the chloride bicarbonate exchanger on the basolateral membrane. So if I showed you a cartoon that looked like that, you would already know that you were in the, that you weren't in the proximal tubule, you were in the loop of Henle, the ascending limb of the loop of Henle. And if I said that this was water impermeable, you would know even more that you were in the ascending limb of the loop of Henle. So I added some information beyond what Claude was asking, but did I answer your, this is a co-transporter because the things that are being transported are moving in the same direction, and this is an exchanger because the things that are moving in opposite directions.
So do we need to know all the exchangers and co-transporters in each part of these different tubules?
Not all, but I think when I went through it, I asked you to circle and highlight the ones that I do want you to know. And so I would ask you to refer back to your notes from class, or to use the key concepts list that describe what is happening. So for example, in the key concepts list, it goes through it by, it doesn't go through it by segment, it goes through it by solute. So for example, when it talks about sodium transport mechanisms, you do want to know that there is a sodium glucose co-transporter on the apical membrane. And you do want to know that there is a sodium proton exchanger on the apical membrane. And you do want to know that in the loop of Henle, there's a sodium potassium 2 chloride co-transporter on the apical membrane. So use the key concepts list to know which of these you do and don't need to know, but also use use the highlights that we went through when I went in class to help you identify which you do need to know.
Because it is crossed out.
If it is crossed out, you don't need to know it.
A question about bicarbonate excursion and reabsorption.
Okay.
In the key concept list, like there is steps to go over.
Okay.
Or to increase pH or decrease pH. And I think it's related to homework too.
Yes, it does, because of the diamox question. Sure. Be happy to do that. Let me just pull up PowerPoint. Okay. So here is the story of bicarbonate excretion. Bicarbonate is freely filtered, and if it is not converted into carbon dioxide by carbonic anhydrase, it will be excreted. Carbon dioxide, which comes from the rest of the body, is converted, let me back up. Let me start the story over again. Bicarbonate is freely filtered. So there is bicarbonate in the tubular fluid, the urinary fluid. And usually, even in the presence of the carbonic anhydrase inhibitor, some carbonic anhydrase is functioning so that the bicarbonate is combined with protons, making carbonic acid, which is converted by carbonic anhydrase to CO2. And the carbon dioxide can go into cells. It can go into this cell, but it's the same as the carbon dioxide from the cell above going into this cell. Okay? So this carbon dioxide can come from here, but it can also come from here, because there's carbon dioxide from the metabolism of the cells around it. Carbon dioxide is converted by carbonic anhydrase into carbonic acid, which splits into bicarbonate and protons. This process, this bicarbonate, is transported by a sodium bicarbonate cotransporter and a chloride bicarbonate exchanger across the basolateral membrane, where in many, maybe most, segments of the nephron. This proton, regardless of where it came from, this proton can be transported across the apical membrane by the sodium proton exchanger in most segments of the nephron, and by a proton ATPase in the later segments of the nephron. Okay, so let's try to tell the story of bicarbonate excretion. Okay, or let's go. Should we go to the Diamox question, to the problem set question?
When your pH is higher than normal.
Okay, pH is higher than normal because of respiratory alkalosis. Right? Jasmine, did you want to clarify something?
I had a question.
Okay.
Okay, you said the HCO3, where does that go? It goes out via a co-transporter?
With this one here?
Yeah.
Yes, there are two mechanisms for transporting this bicarbonate out the basolateral membrane. A sodium bicarbonate cotransporter and a chloride bicarbonate exchanger.
Okay.
Two separate proteins that do this function, which is transporting, pumping, really, bicarbonate from the cytoplasm into the interstitial space where it is picked up by the capillaries. Okay, so let's use Diamox, okay? So, and let's put up a, let's put up a, you know what I'm gonna put up. Put up a Davenport diagram. Let me actually make it good looking. Can everybody see if I draw over here on the side of the book? Okay, I'll get us started. Blood pH 7.4. Blood plasma concentration of bicarbonate in millimolar, 26 or so. Here we are at normal. Here's the normal PaCO2 at normal is 40 millimeters of mercury. Here we are at normal. Respiratory alkalosis, hyperventilation. go to a new curve. PaCO2 is low, less than 40 millimeters of mercury, because we are blowing off too much carbon dioxide in the effort to try to get more oxygen in a high altitude situation. So here is respiratory alkalosis. And now we're going to unpack this arrow, the renal. So this is respiratory alkalosis. This is the renal response, compensation. I've used the word compensation, so let me just use that word. Compensation. Excreting more bicarbonate. So the kidney has excreting more bicarbonate and retaining more acid. So how does the body do that? The body does that by down-regulating transporters and enzymes. When you go to high altitude, you feel ill for a couple of days, and then you get better. It takes days to recover from altitude sickness, unless it becomes critical, in which case you go to a lower altitude because you're going to die otherwise. But let's just take normal response to high altitude. You don't feel well, you feel out of breath, you are in respiratory alkalosis, and then over the course of a few days, your body recovers and excretes more bicarbonate in order to bring your pH back to normal, to help you feel better. So your body has mechanisms to excrete more bicarbonate. And one of those mechanisms is to downregulate carbonic anhydrase, downregulate the expression of carbonic anhydrase. So let's see how that would help. If we downregulate the expression of this carbonic anhydrase, then, and remember this carbonic anhydrase is not in the fluid, it's attached to the apical membrane. If we decrease the activity, the number of molecules or the activity of those molecules, then less bicarbonate is converted into CO2, and more bicarbonate stays as bicarbonate and is excreted. So right there, it's easy. The filtered bicarbonate is excreted, and we excrete more bicarbonate by downregulating carbonic anhydrase. So if we look, right, that's pretty easy right there. Okay, let's look at downregulating this one. If we downregulate this carbonic anhydrase, we produce less acid. We produce less acid that is excreted. So more acid is not excreted. We retain acid. And that's acidification from a alkalosis event. So other things happen. I don't want to pretend that the renal response, the renal compensation to respiratory alkalosis is only a downregulation of carbonic anhydrase. It's not only that. It's accompanied by a downregulation of the sodium proton exchanger. It's accompanied by a downregulation of the sodium bicarbonate exchanger. But all of those things work like downregulating carbonic anhydrase here and here, which in essence allows us to excrete more bicarbonate and not excrete more acid. So we excrete more bicarbonate and we retain more acid moving along this line. And the pH normalizes. And our brain feels better. Diamox is a drug that does the same thing. The drug acts faster than a genetic change in gene expression. So Diamox, by inhibiting carbonic anhydrase, is doing what the body is going to do faster. In fact, people who are sensitive to altitude change often take Diamox prophylactically. They'll start taking Diamox a couple of days, maybe a week, before they go to high altitude, in order to prevent the symptoms from even starting. That's just for your information. So if if you've gone to La Paz, Bolivia, and which is at, I don't know, some crazy altitude, 19,000 feet or something, and I mean, even if you have any experience with having altitude sickness, and you say, boy, I'm going to go to La Paz, I think maybe I should see my physician and see if he or she is willing to give me Diamox before I go. That would be a reasonable question to ask. Anyway, so that summarizes bicarbonate excretion and the alterations of bicarbonate excretion in the setting of respiratory alkalosis, and why Diamox helps us do renal compensation by accelerating the inhibition of carbonic anhydrase. Diamox is also a diuretic because by inhibiting the reuptake of bicarbonate, by increasing bicarbonate excretion, which in fact is accompanied by sodium, so we're increasing sodium bicarbonate excretion, it's a solute. Increasing a solute excretion increases water excretion, which is what diuretics are for. So that's pretty much the whole answer to, I mean, in a nutshell, to that question on the problem set. Did that work for you?
So out of the four places, the proximal tubule, the thick assembly, the cortical collecting tubule, and the cortical collecting tubule. So the first, the proximal tubule, the water is leaky, but the other places the water is not leaky.
Well, let's go through it. So in the proximal tubule, it is definitely leaky. So the reabsorption of solute, especially sodium, but also glucose, is accompanied by the reabsorption of water. And so the fluid stays isotonic. The proximal tubule is a leaky epithelium. So we reabsorb a lot of water as we reabsorb solute in the proximal tubule. The thick ascending limb is not leaky. So in the thick ascending limb, we do not reabsorb water as we reabsorb solute. And that helps us use the Lupo-Pendley countercurrent multiplier. That's part of the component that builds that solute gradient in the medulla, in the medullary interstitium. The distal tubule is complicated. So let me skip the distal tubule for a minute, and then let's go to the collecting tubule. The collecting tubule has variable water permeability. In the setting of needing water reabsorption, that is to say you're dehydrated, the water permeability of the collecting tubule is high because of vasopressin. If you are fully hydrated, the water permeability of the collecting tubule is low because of low vasopressin. Let's go to the slide of, yeah, this one here. So in the distal tubule, oh, that's right, it's not complicated. I'm sorry, I made it seem like it was going to be complicated, and it's not. The distal tubule is not water permeable because the distal tubule receives a hypotonic fluid from the loop of Henle and keeps it hypotonic until it reaches the collecting tubule. So that when that hypotonic fluid reaches the collecting tubule and there is no water permeability, low vasopressin, we produce a hypotonic urine. On the other hand, if there's lots of vasopressin and we have lots of water permeability because of lots of vasopressin, lots of aquaporin, skipping a bunch of steps, lots of water permeability, then the dilute, the hypotonic fluid that was given to the collecting tubule becomes hypertonic because that fluid going through the collecting tubule is going through a very concentrated medullary interstitium. I'm saying the story without showing you the pictures. I apologize. Well, maybe I can just use this picture right here instead of searching for it. The ascending limb is water impermeable, and the countercurrent multiplier of the ascending limb is what built a hypertonic interstitium in the deep medulla. The the distal tubule is water impermeable also, so that the hypotonic fluid generated by the ascending limb of the loop of Henle stays hypotonic, and then in the absence of vasopressin and the absence of water permeability, stays hypotonic until it is excreted. In the presence of vasopressin and the presence of aquaporin expression, this dilute fluid is starting to move through a very concentrated interstitium. Water flows from the dilute fluid to the interstitium, and the fluid becomes hypertonic. It becomes as hypertonic as high a concentration as the loop of Henle was able to build, and we produce a very concentrated urine. So once again, I took Kwa's question and I went overboard with it. High water permeability, low water permeability. I'm sorry, no, got it wrong. High water permeability, low water permeability, low water permeability, variable water permeability.
If the tubule has a high water permeability, does the part have tight active urine?
Tight what?
Tight junction? Like tight
So there's two ways that water can flow. Water can flow, so in the proximal tubule, which is not aquaporin dependent permeability, the water flows paracellularly. It's leaky because there's leaks between the cells. In the collecting tubule, the water mostly flows transcellularly through aquaporin channels. And that's why we can control it readily by the presence of vasopressin. So let me put up the slide I'm thinking of. Here is a cartoon of a epithelial cell in the collecting tubule. It's from that book that includes more information than you want, and the fact that it doesn't include water permeability between the cells makes me, I infer from that most of the variable water permeability in the collecting tubule is transcellular due to water flowing through aquaporin channels on two membranes, apical and basolateral. So in the proximal tubule, the constant water permeability is paracellular, and in the collecting tubule, the variable water permeability is transcellular.
So, is it paracellular for the distal tubule as well? Or
Well,
not the distal tubule, but the descending tubule?
I think so,
Yes.
because it's constant. So it's very likely, I think the only part of the tubule that has highly aquaporin dependent water permeability is the collecting tubule. I think you can safely say that. That said, now I want to double check this slide. It doesn't include the ascending limb, so that again makes me think that it is paracellular in the ascending limb. In the distal tubule, well, we don't have water permeability, so we don't have to worry about it. This slide doesn't go into water permeability in the collecting tubule. And it doesn't go into water permeability in the ascending limb because it's tight and there isn't ascending limb, remember, is water impermeable. So I'm pretty sure that the segments that have constant high water permeability, the proximal tubule and the descending limb, it's paracellular.
Glomerular filtration fit in this thing. I'm just forgetting. So did the height go in the distal tubule?
So you're confused, you have a question about the control of glomerular filtration?
Yeah, does it, like, I'm just wondering why this PowerPoint slide is.
Why did I include that PowerPoint? Let me see. As a reminder that glomerular filtration is controlled in the setting of dehydration. So it's not just vasopressin. So what are the three main responses to dehydration? The first one is vasopressin-dependent increase in water permeability in the collecting tubule that we've just been talking about. The second one is aldosterone-dependent increase in expression of the sodium transport mechanisms in the ascending limb, allowing the loop of Henle to build a maximal concentration in the medullary interstitium. Remember, more more solute transport in the loop of Henle, a more active counter-current multiplier mechanism, a more concentrated medullary interstitium, more of a solute gradient that the vasopressin aquaporin-dependent water permeability can use to remove water from urine back into the kidney. So two mechanisms. Cycle back. Vasopressin-dependent water permeability in the collecting tubule. Aldosterone-dependent expression of sodium active transport in the ascending limb of the loop of Henle, allowing maximal solute concentration in the deep medulla, and a decrease in glomerular filtration rate due to the sympathetic nervous system constriction of the afferent arterioles and angiotensin-dependent constriction of the afferent arterioles. Those are the primary things. We don't have to get into that tubuloglomerular feedback if we know that all we need is angiotensin. But there is tubuloglomerular feedback that produces renin, that produces angiotensin. Yeah, I can do that again. Sorry. So, angiotensin is the third component. Well, so maybe there's four components. Vasopressin-dependent water permeability, aldosterone-dependent sodium transport, sympathetic nervous system, epinephrine-dependent constriction of the afferent arteriole, and angiotensin-dependent constriction of the afferent arteriole. The angiotensin-dependent constriction of the afferent arteriole in part comes from renin. Remember, renin is a hormone that is secreted by the macula densa juxtaglomerular cells. And that renin, there's a whole story. Renin converts angiotensinogen. Angiotensinogen is converted by angiotensin converting enzyme to angiotensin 1. No, sorry, let me do that again. Instead of doing it, let me try to do it on the board, because, okay, so renin cuts angiotensinogen to make angiotensin 1. ACE cuts angiotensin 1 to angiotensin 2. Angiotensin 2 constricts the afferent arteriole. That's the renin angiotensin story, which is a component of the renin angiotensin aldosterone story, but we already talked about aldosterone.
Can you continue that until the GFR part?
Constriction of the afferent arteriole? How does that decrease GFR? Okay. Here's a pretty decent cartoon of the glomerulus, the renal corpuscle. So here's the afferent arteriole, here is the macula densa and the juxtaglomerular cells that produce renin and actually put renin into the bloodstream. Anyway, if we constrict the afferent arteriole, either because of epinephrine or because of angiotensin, then if we constrict the inflow tube, then there is less pressure, less hydrostatic pressure in the glomerulus because we have constricted the inflow. And let's just say we haven't constricted the outflow, although maybe that's constricted a little bit too. But if we constrict, limit the flow of fluid in, lower the pressure, hydrostatic pressure in the glomerular capillaries, that lowers filtration pressure. Lowering the hydrostatic pressure in the glomerular capillaries lowers the pressure that pushes fluid into Bowman space, and that decreases glomerular filtration.
Just one point of clarification. So for the aldosterone story,
Yeah.
what channel is it? The sodium potassium gluchloride channel? Is that?
It's the co-transporter.
Okay.
It's the sodium potassium 2-chloride co-transporter is its expression is highly linked to aldosterone levels and the sodium potassium ATPase, both of them. So then it would make sense that both of them are upregulated in the presence of aldosterone in the ascending limb of the loop of Henle. So the ascending limb of the loop of Henle has the aldosterone receptors that then change the transcription of both of those genes.
When we memorize the cheek R3 and blood pressure 3 through intake and eating, we have to separate because this is the one story angiotensin to J lower GFR, and there is also another story for increasing blood pressure.
Right. So increasing blood pressure overall would increase glomerular filtration, which can also be due to angiotensin. So now things get complicated and things get to be a balancing act. And so in the setting of dehydration, the body is trying to maintain blood pressure by vasoconstriction, but is also trying to limit water loss in part by vasoconstriction of the afferent arteriole. So now we have things are not working perfectly in sync not working perfectly in alignment for the kidney, but we need to maintain blood pressure for the brain, for example, right? And so blood pressure goes up even though glomerular filtration goes down, because constriction of arterioles systemically. Remember what equation am I thinking of? I'm thinking of systemic arterial pressure is equal to cardiac output times systemic vascular resistance. If I constrict the systemic vasculature, then I increase systemic vascular resistance, which increases mean arterial pressure, which we want to do to maintain blood flow everywhere, especially the brain. Mean arterial pressure by itself would increase glomerular filtration, which is not what we want to do. But by constricting the afferent arteriole, we prevent increases in glomerular filtration. In fact, we can constrict that afferent arteriole so much that we can reduce glomerular filtration, even though blood pressure overall, blood pressure measured in the arm, not in the kidney, is high. So glomerular pressure, even in the setting of dehydration, glomerular pressure will be low, even though systemic pressure is trying to be maintained or even high because of that equation. Another reason, so I didn't really teach this, and so I wouldn't test this, but this might make sense. In the setting of dehydration, I'm not going to repeat all the things I've said already, because you have them. But if there is a low glomerular filtration rate, we now understand, then there is low flow through the tubule, because there's low fluid being filtered means low flow in the tubular fluid. If the rate of flow through the tubular fluid is low, then the countercurrent multiplier system can work more efficiently. And that also helps to build this high concentration solute in the medullary interstitium. Okay? So they're all working together. Low glomerular filtration, high solute transportation here, low flow here, buildup of the gradient, high water permeability so that what is put out is the same osmolarity as the gradient we built to produce urine, which we have to produce, but it's got a high osmolarity, which means it has a relatively low water content so that we're conserving water. Well,
do you mind going over the relationship between, it's the same slide, but like vasorector, kind of like compensate for the glucosamine?
it doesn't compensate for the loop of Henle. What it does is prevents the blood, the fact that it's making the loop in parallel with the loop of Henle prevents the blood from washing out the gradient. So you're thinking of this slide, which is nasty. So let's look at this slide instead. Let me try this. See if this helps. So the countercurrent mechanism of the loop of Henle, let's just say it's working at maximum, has built up a 1200 milliosmol interstitium. And after all, we're at 300 milliosmolar here. So there's the gradient, that pretty much the maximal gradient we humans can build. Desert rats can build up a gradient that goes from 300 to 3,000. The vasorecta, by paralleling the loop of Henle, allows water to be reabsorbed from the vasorecta, from the loop of Henle. The vasorecta, by paralleling the loop of Henle, allows water to be picked up by the vasorecta from the loop of Henle without maximally diluting this gradient that the loop of Henle spent a lot of energy building. So, and it's hard to illustrate how that could be. But let me put up a thought experiment of what would happen if we used this kind of capillary network, you know, sort of your normal mishmash capillary network here. Well, let's just do that. So if we had a capillary that is sort of wandering around like that, going, you know, coming in this way and going out that way, right? Just a capillary coming off of an artery, crossing. Remember, what's in this capillary is 300 milliosmoles. And the situation on the outside of it is 300 milliosmoles. No problem. Okay, so not an issue. But if I took a capillary that did the same sort of thing down here, and then worked its way up into a vein, we've got a capillary that's at 300 milliosmoles in contact with a fluid that is 1200 milliosmoles. Water is going to flow from that capillary into that 1200 milliosmol fluid, diluting it, washing out the gradient. By paralleling, rather than transecting or whatever, going, you know, random, we minimize that washout. Because every part of a capillary has an osmolarity that has already been sort of adjusted for being in contact with that high osmolarity fluid. And there's just less washout. So does that help explain that situation?
So I think in the previous class you said there's a little bit of difference between those polarities and not exactly the same.
That's
amount
right,
of water.
because there is water being reabsorbed.
Okay, but it's not significant to wash out any.
The idea is that we want to reabsorb water without washing out the gradient. And that's another reason why you could say, well, in the collecting tubule, in a dehydration state, there's water flowing out here, which would dilute the gradient. But remember that now we have urea, so we have water coming out and urea coming out in more or less the same amounts. And urea, so what's coming out is not diluting the solute gradient as much as it would be if we didn't have urea. The main point is in a dehydration situation, so I'm going to erase this now because it's wrong. Don't want you to get the impression that this is the way it is. This is the way it is not. The point is, the loop of Henle used a lot of energy to build up this gradient. And we want to use it, and using it means we have to wash it out a little bit, but we don't want to wash it out a lot. Me?
When you mentioned dehydration, like where in the body would we want to conserve water the most? Like either institutions or in the blood capillary?
Yes, everywhere. Right, so we need to maintain an interstitial volume, especially in the brain. We need to maintain an extracellular volume. We need to maintain blood volume for blood pressure. We need to maintain cytosolic volume. So where the water goes is everywhere. Where we're conserving water is pretty much everywhere. I don't have an informed answer to that question. I have just a sort of figure it out answer to that question. I don't think there is a part of the body that needs water the most. And I don't think there's a cellular fluid that needs water the most. I think you should just think that it's needed everywhere.
So previously, you mentioned that response to dehydrations are increase of vasopressins independent.
Increase in vasopressin dependent water permeability
Second
in
is,
the collecting tubule.
second is the aldosterone dependent
Aldosterone dependent
in the collecting tubule.
in the ascending limb of the of the loop of Henle.
Oh, yeah. the
Aldosterone is helping the loop of Henle build this gradient.
And the third one being decrease of
Decrease in
glomerular-
glomerular filtration by constricting the afferent arteriole, decreasing the glomerular filtration. Less fluid is filtered. That's helpful for decreasing excretion. If we don't filter it, we can't excrete it. And the other way is by decreasing the rate of fluid flowing through the tubule, we help the loop of Henle build this gradient as well. So the glomerular filtration control is helping us in two ways. We can't filter it, we can't excrete it, and by not filtering it, we decrease the flow rate through the loop of Henle so that it can build the gradient better. Let's take a break. You can confer amongst yourselves and decide if you have additional questions. Yeah, let's take a short break. More questions now. I'm also happy to answer more questions as we're working in the lab this afternoon. I hope you all got my email about lab this afternoon. Some of you are starting, basically, the starting lab on the hour, right? And you have your assignments as to which hour you're starting the lab.
Is the adrenal gland located on top of the kidney?
Yes, the adrenal gland is sometimes called the suprarenal gland, which means superior to the kidney, which means above the kidney. So let's see it. Where would it be if I showed the gland? So we didn't see the adrenal gland in the cadaver because I think when we pulled out the kidney, we left the adrenal in the body. But in this cartoon of the body, these yellow structures superior to the kidney, suprarenal, are the adrenal gland. This figure makes it look like it's fat. It's not fat. It's renal gland. There is renal fat, but this cartoon, yes, above the kidney. Short answer.
But the process of cutting the angiotensinogen take place in the liver, right?
So angiotensinogen, so I won't ask you questions about angiotensinogen yet because that's a liver function, which is the next section of the course. Angiotensinogen is produced by the liver and released into the blood. Renin is produced by the juxtaglomerular apparatus and released into the blood. The reaction that renin catalyzes, the cutting of angiotensinogen to angiotensin 1, happens in the blood. It happens everywhere in the blood. Angiotensin converting enzyme is an enzyme that's actually produced in many areas of the body, including the lungs. So when that blood goes through the lungs, one of the things that happens is angiotensin 1 is cut by the ACE in the lungs to angiotensin 2. So I didn't cover that in the respiratory function because that's not exactly respiratory physiology, but that is something else that happens in the lungs. This is not something that you need to know for this course. Yeah.
So there was a tubular glomerular defect,
Okay.
and so we kind of went over it, kind of covered the sympathetic nervous system through the neurotransmitter release.
Yeah, that one's
and the
easy.
constrict-
Yeah.
Yeah, but I feel confused with the concept of tubular glomerular defect.
Yeah, you and me too. So let's see if we can figure this out together. I'm trying to find the perfect slide. Let's do this one. Okay, this is two superiors, two below generator. In some ways, the thing that's confusing is that I put tubuloglomerular feedback in the same context as sympathetic nervous system the response to dehydration. Tubuloglomerular feedback is a sort of general purpose feedback mechanism to control GFR and to control the rate at which fluid is flowing through the tubules. So it is not only in the context of dehydration, it's also it's in general feedback mechanism. So keeping that in mind, that this is not specifically a way that we control glomerular filtration in the context of dehydration, but it's a way that we control glomerular feedback in general. Let's go through it. So the idea is, if there's so I've skipped some steps that are most confusing because we don't really need to get lost in those weeds. No, we could try it again, but if there is high flow in the distal tubule, which is not what happens in dehydration. But let's just say the kidney is saying, this flow is too high. It doesn't allow me to build the concentration gradient in the loop of Henle that I need to build because the flow rate is too high. Let me shut down the flow a little bit, or some, by decreasing glomerular filtration. Just in general, not in the context of dehydration. So there is high flow in the macula densa, release of ATP from the macula densa in response to that high flow, a series of events that lead to constriction of the afferent arteriole to reduce the flow, right? So it's just simple, it's a simple-ish, maybe too simple for us because we're thinking about dehydration or hyperhydration. In this case, we're just talking about controlling the flow rate. If the flow rate goes up, we want to lower the flow rate. If the flow rate goes down, we reverse all of this and we make the flow rate go up. We want a baseline control of flow. That's what tubuloglomerular feedback is all about. Morgan?
This feels like not the most relevant question, but it's kind of a real life application. People always say that if you drink water, like if you drink half a liter of water in smaller amounts over the time period, it's like better for rehydrating than if you would drink a bunch at once. Can you myth bust that?
I don't know. Not off the top of my head. I'd have to read what people are saying and try to think of, I mean, so I could come up with a plausible way to not myth bust that, to actually make that seem plausible. So if you drink a half a liter of water, you chug a half a liter of water, the osmolarity of your blood is going to go down pretty quick, because your stomach will absorb that water pretty fast. So the osmoreceptors in the hypothalamus are going to detect a sharpish drop in osmolarity. And they're going to say, whoa, let's shut off all vasopressin. And then we'll immediately produce as dilute a urine as we can produce. Whereas, you can imagine if you step the water level, the hypothalamus isn't being hit so hard with a change in osmolarity. You can imagine that the hypothalamus might not maximally shut off all vasopressin, and you might conserve water over that period of time a little better than if you chugged the half a liter and produced a maximally dilute urine. So that's I don't know if that's the correct way to be thinking about it, but the brain responds to changes a lot more than it responds to, in general, this is way oversimplification, but we respond to changes more acutely than we respond to levels. And we respond to levels too, but we also respond to changes even more. And by changing things fast, we might get a more active response for producing a dilute urine than if we change that hydration slowly. So I don't know if it's true, but it wouldn't surprise me if it were.
So in the problem set, just by looking at the number, I can tell that the concentrations of potassium is higher in the plasma, lower in the plasma and higher in the urine. Is that because the volume of water in the plasma is higher than that?
So what numbers are you looking at?
Concentrations of potassium in the plasma and in the urine.
Oh, you mean the numbers I actually gave you in the question? If you look at the concentration of potassium in plasma is 4.5 milliequivalents per liter, and the concentration of potassium in urine is 100 milliequivalents per liter.